tiny biological production factories ever give petrochemicals or animals a run for their money when it comes to unit economics?
We are getting very, very close. And I think you're right, we've been through a cycle of hype. Today it's more realism and conservatism that's affecting the industry. I think that's a good thing. I think we've generally underestimated as an ecosystem what it takes to bring truly affordable, scalable and desirable biomanufactured products to the market. But now companies are much better aware of what it takes, deploying capital in the right way, forming the right partnerships to make it happen. So I'm very, very optimistic that we will see the full promise of the bioeconomy and biomanufacturing come to fruition in the next decade.
At the moment, do you feel like precision fermentation only makes sense for very high value products and ingredients? We've got companies making HMOs or lactoferrin, things like that. What about more commodity kind of ingredients like, BLG you work with, or casein?
Mm-hmm. I think we're absolutely seeing that today the bulk ingredients are enzymes and pharmaceutical products, and then it's making its way into the true bulk ingredient space, and we're really excited about some of the technology developments that are being secured these years, where 21st Bio is among the leaders, and there are certainly others too, where ovalbumin, BLG, caseins, alpha-lactalbumin are starting to come online at relevant price points. I think we will see in the next couple of years that we will unlock below 15, even below $10 a kilo for some of these ingredients, which will bring us into the price range where we can long-term compete against animal-derived protein.
What are some of the biggest challenges that startups who are trying to cross that valley of death are facing?
I think what many startups are forced to do is to use the strains that are readily available, either from academic labs or from some of the suppliers that are out there. And what they're typically selected for is speed in their ability to be manipulated. So they're really good for proof of concepts. But once you want to take them into industrial scale, where the conditions are very, very different than they are at lab scale, these strains are no longer thriving. And so their performance tends to decrease significantly. And it makes the iterations going from lab to pilot to full scale very, very tough, error prone, expensive and time consuming. So I think what we need to hopefully see in the industry is that we rally around a smaller number of chassis strains that are dedicated and optimized for thriving at full scale, such that we can avoid running into this slow iteration, expensive learning loop, but more in an engineering fashion, move predictably from lab scale to full scale production at the same technology performance as we have selected for in the laboratory.
Is the idea that if, say, a startup works with you, that they can work with you and get to the stage where they're ready for that tech transfer stage to work, say, with a CMO or a larger company?
That's exactly right. Yeah. So we don't produce ourselves. We don't pride ourselves with being masters in discovery. So companies that we work with will have to do their own discovery, their own application understanding, and tailoring their solution to the market that they're going into. But we will then prepare the manufacturing technology from strain to fermentation to downstream process if they want us to, and then tech transfer that to a CMO of their choice, or if they have access to own internal production. And we think that can be the equivalent of how computers were developed in the beginning, where lots of different software were running on other people's hardware, and very few, if any, were trying to cover the whole stack simply because of the complexities that they're facing. And I think biology needs to return back to some of those same principles and distribute the labor and the development work that's required to bring about some of these innovative solutions that we all need.
In terms of some of the challenges, you talked about that kind of productivity issue, and as you kind of scale up, it doesn't necessarily work in a linear fashion. But one of the other things a couple of people have said to me at this conference is that disconnect between work upstream and then downstream. So you might have two teams that aren't really kind of talking to each other, and you'll have something that will increase, will be beneficial upstream, but then the net effect of that is that you can maybe recover less of the product downstream. Does there need to be more of an end-to-end conversation?
I fully agree. these things cannot be developed in a sequential manner.
Yes.
Sometimes you need to do it because of resource constraints or team size constraints, funding constraints. But in the big biotech companies such as Novo Nordisk and the pharmaceutical industries, all of these things are happening at the same time, and you are, so to speak, iterating from end to end. And so you get to a first generation strain, you start optimizing the fermentation at the same time as you're optimizing the DSP. It's the only way to win. But it has to be done from a mindset of striving for simplicity, to your point, such that you don't introduce complexities in one stage that will make it impossible to succeed at another stage. So getting to this notion of platform technologies, platform strains and recipes, where you're reusing the same basic principles, maybe with slight modifications depending on the product you're producing, can alleviate a lot of that complexity and risk where decisions in one team is negatively impacting decisions in other teams.
So you've got companies who you might see at SynBioBetter or even at this conference with kind of enabling technologies that they claim could sort of change the game in biomanufacturing. Are there any particular ones you're monitoring, whether it's continuous fermentation or optogenetics or being able to more rapidly domesticate wild microbes?
Obviously, continuous fermentation is a hot topic, and it's been around conceptually for a long time. We are going to the market with batch fermentation for the first couple of generations just because it's tried and tested, but there are some potentially really, really attractive advantages of transitioning to continuous. We do need to see it move up in scale such that we are also confident that the benefits that are currently maybe at the 10 to 15,000 liter scale can be replicated in a 200 or even 400,000 equivalent type facility. But I'm sure that will sort of make its way, and in due time that could be a really, really interesting technology to tap into. I think AI is bringing about a lot of opportunities, both in discovery, where we can tap into new protein functionalities and certainly more directed designs to targets or functions that humans want but nature didn't need, but also in the way we engineer the strain and make sense of its metabolic mysteries to direct more of its efforts towards protein engineering. I'm sure AI will help us understand the data sets that we can already generate cheaply, but perhaps not get the full understanding of. So
what about feedstocks? Because again, at events like this, you often hear people say, oh, the economics of this will only make sense if we can have a broader range of cheaper feedstocks, whether you've got companies doing gas fermentation and so on, but also kind of upcycle waste streams and so on. Is that going to be important for the next generation of products, and also finding then strains that can utilize those different feedstocks?
Yeah. I think it's a really, really good point. For the first generation of products, I think glucose can get us where we need to be. But I think when we are thinking longer term, and certainly we're thinking about local production of some of these food ingredients, for example, or ag solutions or other things, then we need strains and processes who work for the substrates that are locally available, whether that be glucose or sucrose or even lactose. And we believe that the same strains, the chassis strains that we are using today, can absolutely be adapted to some of these carbon sources. But there are, of course, some performance drops that we're seeing today that need to be closed, and that will take development effort. But it is the way that we can regionalize and localize biomanufacturing, which I think will mean that many communities can take back control of their nutritional challenges and also their supply challenges, for example, for biopesticides or bioherbicides, who can help them sustain local production for food and agricultural products.
What I'm hearing again from some people in the industry is that if they look at available kind of co-manufacturing facilities, for example, it might be that they've got what you want, but not exactly what you want on the downstream side, or they don't have a spray drying on site, things like that. So is that kind of a challenge?
It is a challenge for sure. I think it's especially a challenge in the realm where each company is going for their own strain and their own process. If we were to unleash this vision of transferring to platform technologies and strains where we can reuse the same DSP and so on, this challenge would become less. But certainly today we have a couple of these facilities around where some investments are needed to unlock the potential before they can be put to use.
Delana is here, big LATAM kind of sugar company, and they have got this biochem division, and they're talking about building a large precision fermentation facility in Guatemala. Are those kind of developments encouraging for this industry?
Very much so. I think that is exactly the kinds of partnerships that we want to see happen. Whether they choose to use our strains, their own strains, I think matters less. Each can do their decision, but the fact that facilities are being planned, are coming online, are built in vicinity of the raw materials that are needed to run them, is an amazing thing. Once capacity is there, we can quicker deploy the technologies as they mature and get products to consumers fast.